when you got that team together, the company's humming along, you're developing this technology, demonstrating its value. What's that, I guess, sales proposition? I don't want to say sales like in a salesy kind of way, but sort of that value proposition you go to the industry with and you say, AI, our application of AI that gives you valuable, like actionable imagery is better than AI that spits out prose, for instance. Lay that out.
It's like you are kind of asking a question about words and images, and you mentioned AI. Okay, so that's actually an interesting question, because we are seeing today AI coming into two different worlds: the world of words and the world of images, and they actually kind of came onto the scene almost at the same time, with generative AI for language and generative AI for images. And actually, when you look at it, you could say that so far, and maybe I can explain, I think, why, AI is actually better in images than words. And the reason that this is the case is that, actually humans are like that as well. If you think about it, words are ambiguous. Images are just data. It is what you see, okay? Yeah. Then you come to explain it, then you need the words, then different people will describe the same image in very different words. So basically, if you go and train AI on words, it gets all that ambiguity into it. So you train it on 1 million words, and it has millions of ambiguities. If you train it on millions of images, you are just giving it millions of examples of the thing.
Yeah.
So actually, if you will actually look at this in the context of applications, for example, if you try to explain what is the difference between a dog and a cat, and put it in language as rules, you will run out of paper space before you figure out that for every rule, there is an exception. And you cannot actually say what is the difference. But when you see a dog or a cat, you know the difference. You know if it's a cat or a dog. Now, AI will find it very hard to explain to you the difference between a cat and a dog. But give it 1 million examples of a cat and a dog to a neural network that is seeing those images. Give it now the 1,000,010 image and ask, is it a cat or the dog? It will immediately classify it correctly. So this is a very robust application of AI. The most robust application of AI actually is the ability to see in an image something and classify it versus it A or B.
Yeah.
Now, why is that even relevant to all we're talking about?
Yeah, maybe I was thinking that maybe I should have backed up a little bit and said, okay, let's talk about the AI aspect of Anima's solution before we got into the value of the imagery. But yeah, as you were saying.
So actually, yes, it's like, why do we need AI in biotech? Okay, maybe we should even start there. Okay, what's missing? What was not working?
Well, there's so much discussion around AI and biotech, and so many levels of application of AI and biotech that I think there's a lot of misunderstanding out there, or I don't know if it's misunderstanding or just a general lack of understanding around the real killer apps, like the real potential of AI. So
yeah, this is exactly, I would say that this is, this comes to the question of what is AI really good at that we are not. Because if you want to bring AI, it has to have some value, and there is something that it can do that we cannot do. So it is really, when you try to purify this and to understand, it's about the ability to make sense of a lot of data.
Yeah.
And this is what AI is really good at. What people are not good at is to make sense of data, we are good at. And AI is actually sometimes struggling to come up with reasoning in quite simple situations. But when it's a lot of data and you try to make sense of it, you can't. So biotech is all about biology, and biology is a lot of data. And it's unrelated data that people are trying to connect together. So there are millions of publications about diseases and proteins and roles and targets and molecules and drugs. And it's extremely challenging just to understand the links between all of these elements. And you are biased towards what you know and what you have read. You don't know what you haven't read. So you can now use AI to summarize those things for you and find connections and discover mechanisms by kind of connecting that if A is related to B and B affects C, and C is a protein that is doing something with D, then there is a connection between A and D. And it's good at doing that, and it summarizes this into knowledge graphs that you can visually see it. And this is actually a powerful way to look and learn about diseases and mechanisms and targets that could be involved in diseases. But notice one thing, okay? And this comes again to the words versus the images. These are words. It is good in connecting the words when there are many, many words, but it is starting with the ambiguity that is in the words themselves.
Right.
So these publications have ambiguity. The data is incorrect. The assumptions are incorrect. It didn't understand what it is being said really. Eventually, the conclusions are not what is said in the abstract, but the abstract is selling the article, so that's kind of forward-looking statements into what could be done, but the experiment didn't prove it, and everybody is citing it back then afterwards as something of effect. So AI actually is good in images, and images are a completely different way to approach the whole problem. So if you're looking at experiments, all these publications, by the way, are after the fact summaries of priorly done experiments.
Right.
But those are very biased because of the words and because of the connections. So each of them may be good, but many of them are not. And then the connections are wrong. Now imagine that you could do it in a completely different way. And this comes actually later to what we have done also at Anima. If you're doing an experiment and you image it in images from inside the cells, looking at the biology at play, you are basically observing the real biology.
Yeah.
And most experiments really, those in publications, for example, they are measuring one parameter, okay? But if you could actually visualize an image, just like we people visualize a cat or a dog, we visualize at once 1,000 parameters, 100,000 parameters. We cannot even say what the parameters are. We don't think like that. We just visualize it. So imagine that you could generate millions of visualizations, for example, a disease like Alzheimer's. You take and you visualize 100 million cells in parallel at once, automatically, cells that are diseased and cells that are healthy. And now you give it to a neural network with AI to look at that and say, what is the difference? Now, it will not tell you the difference in words, right, but it will understand the difference, just like the dogs and the cat. It doesn't explain what are the rules, but it knows now if they see a cell, is it diseased or healthy? Now, this is huge because now, if you can actually have the technology that is seeing, as we are seeing, the real biology pathways, for example, in mRNA biology, now you could go back to that knowledge graph, so to speak. Yeah. Now you're asking AI a much better question. You're telling him the difference in Alzheimer's disease between healthy and diseased cells, based on 100 million examples that I've seen, is this pathway. Go and figure out for me now, with your abilities, what are all the proteins along this pathway? Is any of them known to be associated with Alzheimer's? And now we are really into understanding the disease mechanism. But AI is more helpful, actually, if you give it real data, unbiased data, 100 million examples of it. This is where the real power of AI comes to play and shines at best.
Yeah, and I don't want to come off as sophomoric here, Yochi, but the skeptic. I'm gonna throw a skeptic's I guess perspective at you. We've played with AI imagery here in the office a bit. Consumer grade, right? So this is where I'm going to ask you for some differentiation, because the perception among perhaps many of our listeners who have only had that kind of consumer grade interaction with Chat GPT or whatever other image based AI tools are out there, on Google, is like, I'm not so sure about the accuracy of the data, because when I ask it to, create a picture of a woman sitting on a chair, oftentimes the chair has 5 legs and the woman has 6 fingers on one hand. So we look at that and we go, well, I don't know if AI is that good at imagery. What's the difference here between the applications that you guys are building and the ensure, I guess, your assuredness around the accuracy of the tool in your application versus, what so many see from sort of a consumer level?
So actually what consumers see today, there are 2 things. One of them is generative AI for images. And indeed, this is the ability to create an image that is a new image. And those images can be stunning, but as you said, in the little details, it could miss. Yeah. But actually, I'm not talking about generating images by AI. I am actually talking about image understanding by AI, given an image that is the real image. It's not an image that AI generated. For example, Tesla is already offering self-driving vehicles. How does that work? And this is working. This is the most robust application actually of AI out there, and it has been working for a while because it comes back to something that the AI is really, really good at. Tesla has cameras that were on the cars, driving for a couple of years on 10,000 vehicles and taking videos, which are broken into images, of what roads and driving looks like. This is a stoplight, this is a road, this is a car, this is a person, this is a tree, this is a sign, this is this is that. You are seeing millions of examples of that, and then it becomes so much information and so many examples that it can actually drive in real time and knowing what's around. I'm talking about something that is extremely simpler than that even, which is, let's look at 150 million images taken from cells of a disease model. Now, these are real images that indeed we take with our imaging technology, but they are the ability to see and visualize the actual biology. It's not generated by AI. It's just taken with a microscope. It's a high-powered microscope that runs in an automated lab and takes 150 million images from all of these single cells. So the data is there. This is the biology. Now we apply image classification and understanding on top of it, just like the cat and the dog. Okay, it's a completely different paradigm.
Yeah. Okay. What does I'm curious that as you describe this, and I'm understanding the value of that visualization of an actual cells. There's no room for ambiguity. There's no room for interpretation that you get with the written word. There's really not even like a sort of a time parameter that skews the data, because it's real. It's potentially real time, I would assume. I mean, the time factor is going to take it off the table. What does the translation of the machine's understanding of what it's seeing to the human being who can do something about it look like? Does it remain visual through the application state, or is it at some point, is it subject to analysis in a visual form, and then decisions are made based on that visual analysis? So I guess I'm wondering like what the action point is at the end of the process.
So let me describe to you our platform and how it is applied, because this is essentially how we are connecting all of these things together and where basically we are bringing it to add value. So really, in a nutshell, our platform, it's called mRNA Lightning. And mRNA Lightning platform visualizes and decodes the mRNA life cycle and is used in multiple applications of drugs and target discovery, RNA drug optimization, mRNA vaccines optimization, anything that relates essentially to, I would say, drugs and vaccines in mRNA, okay? which is a particular specific area in biology, but an extremely promising one and a very big one.
Yeah.
Okay, as we've seen with COVID, for example.
Yeah.
So we have really an automated mRNA visualization screening technology. We generated over the last decade more than 2 billion images of mRNA biology. And this was done in dozens of diseases, cell types, conditions. And we use this massive data set to train disease-specific, what we call AI imaging neural networks. And they are capable of recognizing what we are calling disease signatures. This is really the difference between disease and healthy cells in a disease. And it's a particular mRNA biology pathway because the visualization technology enables us to take 50 different visualizations in each cell. Each of them is a pathway in mRNA biology regulation. So it's something super important in the regulation of the mRNA. Now, what happens now is that once the differentiation between healthy state and disease state is kind of seen in 150 million images, the AI neural network kind of extracts the disease signature, that pathway out of it. It tells you this is the difference. And this is actually a huge step towards answering the biggest question of all, which is why is the disease happening in the first place? Yeah. What's the disease mechanism? What's the underlying disease mechanism? So essentially now, after the screening phase and the generation of that deep data in images, you have an image bank that is almost like the disease atlas, if you want to call it like that. It shows you why the disease is different from healthy state. However, you want to actually do something about it. So this is where the AI analysis of the massive data set actually comes into play and turns it into numbers. It quantifies everything for you, and it analyzes it, and it suggests novel targets. And it enables you to get started with the next phase, which is screening for compounds that would modulate exactly that pathway and will identify for you a drug, basically. So it's a drug and target discovery platform. In the same way, and by the way, this data that is generated and gives you like unparalleled insight into the disease mechanisms, and it aids you in optimization of drugs and discovery and prioritization of targets. The words are coming now back. The world is coming back because into the system, we plugged in a large language model, which is the mRNA Lightning LLM. Now, we are not in the business of developing LLMs. Nobody is, other than a few companies in the world. Yeah. But you can bring them in if you have data that is robust, that is unambiguous. They will be actually very good at that. So we are bringing in LLM, and we've built a chat-based interface through ChatGPT, which we call the mRNA Lightning Copilot of the system. And you can interact with the data in natural language. And all this analysis, screens, and all this information is actually presented by the system. So it's an automated screening now of compounds follows. And we identify hits, compounds that modulate this, and then we optimize them. So it's a full-scale drug discovery and optimization platform. More recently, we developed applications that are for vaccine optimization, for RNAi drugs, siRNA, ASO optimization. And all this platform really runs in a massively parallel automated lab architecture. And the screening is really high content, but it's done in high throughput, like huge data that is generated. And it's finally also an open system. So you can extract all the data to other AI analysis tools in case that our analysis, for whatever reason, is not what you want.
When you mentioned the vaccine and RNA therapeutic optimization additions that are more recent. When you began, I'm just wondering sort of like your strategy or your approach around the development of the platform itself. and you start out, you're like, hey, we could use this. This is a great tool for target discovery or a great tool for molecular discovery, small molecule drug discovery. Um, did the additional applications kind of unfold as you saw the technology having a fit there, or are those things that you thought, well, we need to go chase that down because our mRNA vaccines are a big market opportunity and we need to make our application work there. How did sort of that evolution, I guess, of the platform happen?
So I'm actually looking at things from my previous companies, and there are two things. One is a product, which most people are focused on, and the other one is a market, which most people forget about.
Mm-hmm.
So it's so much harder to succeed in a company where there is no market for what you're doing than when to succeed with a product when there is a market, even if your product is not so good.
Yeah.
So, and our product excels, and we started it in small molecule drug discovery, that screening of compounds that basically are modifying the image as they are in the cells. You kind of turn it into a massive unbiased drug discovery screening campaign by taking, let's say, 300,000 molecules, and each of these, when they are in the cells, if they do something to the mRNA biology, they will change the image. And now we are back to the dogs and the cats, but now we are looking for a molecule that will make the image look more like a dog and less like a cat. So the hits are those that are modifying the images from the diseased to the healthy.
Yeah.
And the AI has a neural network that can tell, given an image, what it looks like more. So it flags for you those that are active on the mRNA biology pathway. So you know that you've got a compound that is active. You even know already almost where it is working. And this is huge. Maybe we'll get to that later about the mechanism of action. Usually that's an after the fact, hard and mostly not successful, unsuccessful challenge. Here, you can get it even before you start. You just are guided to it. So when I looked at this, I said, okay, we've got this working. We did 3 partnerships with big pharma companies, Lilly, Takeda, and AbbVie. We've got that application fully figured out, and we are developing a pipeline of our own. And I was around, presenting the story, talking about it, and all of a sudden, I start to hear people coming to me, and they say, well, we are in mRNA vaccines, and if we could visualize the vaccine, which is an mRNA, inside the cells with your technology, we could actually profile 200,000 different designs, and you can actually visualize which one looks better. Now, and I asked them, what does it mean look better? In my terminology, healthy and diseased. I'm moving from disease to healthy, but here is a vaccine, and they're all the same cells. They say, no, healthy and diseased, switch them for less efficacy, more efficacy. So we want the vaccines that are translating the most protein and in the right place. And we want could you actually find, for us, by comparing all of them, which ones look the best? So let's throw 200,000 into your system, massively parallel environment like this. You screen them, you come back with 10,000, with 100,000, or 50,000, 40,000, whatever the screening success rate is. It will usually be like 20-1 reduction right away. And then you throw them back and you say, okay, permutate a little bit about those, bring them back for a second optimization run, and now you will have maybe only, 2,000. And every time we visualize and compare, and it's a race between the vaccines. Yeah. You can see that this is the same thing, actually. It's the same technology applied to a different problem. Now, siRNA drugs have this problem, which is the endosomal escape challenge. 99% of the RNA, of the siRNA, actually gets trapped in there and never gets into the cytoplasm of the cell. But we see it, we see where it is, so we could do things, that modify and enable you to choose those that are actually best in doing that. So again, it's the same stuff. So if you have a machine like that, A machine, it's not a machine. It's an architecture, a platform like that, and it can visualize the biology inside cells. It's applicable to all these problems. So now we are interacting as potential partners with companies that are coming from vaccines and siRNA drugs, and we are exploring, the value of the technology in partnership mode. We are not going to develop those, but we are going to offer the technology for those problems.
Yeah, very, very cool. You mentioned MOA, and I do have a specific question about that because, as you know, plenty of drugs get approved and go to market. There are drugs that have been in the market and in patients for years and years and years where we still don't have a thorough understanding of that drug's mechanism of action. We know it's safe, we know it's effective, name your drug. So why is it important now? Like, give us some perspective on why this advantage of your platform that gives you early access to an understanding of mechanism of action. Why is that important now when we've kind of been living blissfully ignorant of so many mechanisms of action for as long as we have?
So indeed, about half of the drugs that have been approved were approved without an understanding of the mechanism of action. But this is the old, I would say, game. Right now, if you speak with a pharmaceutical company and you've got a compound and it's a great compound, but you don't know how it's working, then unless you go all the way to clinical trials and validate that it's safe and the efficacy is there, they will not want to touch it. So, and the reason is that the side effects of that compound could be that it touches upon targets that are bad targets that have failed already in the clinic. So the idea is this. Yes, if you have the money and you want to go by yourself all the way to do all the clinical trials, and only there, you will find out, or maybe you don't have to find out, it's just a matter of spending so much money, and maybe you could avoid it if you knew the mechanism of action. Because if the mechanism of action is going through certain proteins, kinase, for example, that has multiple functions, and your drug is hitting that kinase as a side effect that you are not aware of, that's like already known to be downstream big problem, and they will avoid the project altogether. So I've seen that pharmas are actually very, very sensitive to know the mechanism of action of the target before they will actually invest to go to the clinic with the product. You can develop them all the way to preclinical and not know it, but it is hard today to convince people to go for I would say, project without the mechanism of action, and it forces you to go into the clinic almost on your own.
Yeah.
And this is a very expensive proposition, especially if you have a broad pipeline. However, if you look at what is happening today with technology, that's a very, very challenging problem. Given a molecule, how does it actually do its magic? It's a harder problem than finding the molecule sometimes.
Yeah, right.
And how do you solve that? So we used to solve it by a technology that we introduced 3 years ago called MOAI, Mechanism of Action with AI, which was kind of taking a backward look. Here's the molecule, here's the data that we had before during the screening, try to figure that out for me. But that's really challenging still, even for us. I mean, we had great success with it, and in I would say 5 out of 8 projects in the last 3 years, we've done it successfully. That's 10 times better than the success rate of the industry already. Yeah. But what we are doing now with the neural networks and the ability to actually see right from the beginning what is the pathway that is different between the healthy and the diseased cells, it's like already knowing the MOA almost. You are there in the neighborhood of the target. Yeah. So that's a game changer. That's turning the pyramid on its head. It's not starting the project and hoping to find the MOA in the beginning. It's starting the project, understanding what you are going to target, and then actually screening from there. So it's really an interesting proposition. Now, you can do this, as some companies are trying, with words, as we said, looking at the publications, looking at the disease, what is known, come up with ideas for targets. I think that our approach, which is unbiasedly just looking at the biology give the AI 100 million examples, it will tell you the pathway. Now go with that pathway to the LLM and ask it what it knows from publications about it.
Yeah.
It's a much more powerful approach in our opinion.